Hello again, everyone, and welcome to our webinar on the use of QCI Interpret to elucidate precision medicine in breast cancer targeted therapy, especially during this month of breast cancer awareness. My name is Christy Cloyd. I am the Manager of Advanced Genomic Support in Americas, and I'll be your moderator today. In this webinar, we'll explore a few breast cancer case studies using QIAGEN's solution for clinical decision support in oncology called QCI Clinical Insight, or in short, QCI Interpret. Before we start our session, I want to make sure that everyone can see the slides that should be shown on the screen and hear us clearly. If you can't see the slides for any reason, please send me a message through the control panel. Please stay for questions at the where we'll hear more from Dr. Wiranatne, who I'll introduce shortly. Questions will be taken at the very end for as long as time permits. Please submit your questions through the question window in the control panel, and you can do this throughout the webinar. Any questions that we don't get to answer at the end of the webinar, we'll follow up with responses by email within the next week. Our speakers today are Dr. Julie Deschen and Dr. Dilhan Wiranatne. Dr. Deschen is a manager in advanced genomics integrated solutions, and before joining QIAGEN, she studied breast cancer as part of her doctoral work, and then joined Ingenuity Systems and contributed to applications which allowed users to prioritize and rank variants, expediting disease discovery. Dr. Wiranatne is a senior scientist with Advanced Genomic Support Team, and before joining us at QIAGEN, Dr. Wiranatne worked at leading diagnostic companies including Thermo Fisher and N of One, and is an expert in oncology, clinical interpretation, and in the variant annotation space. Again, thank you all for joining. Dr. Deschen, please feel free to begin your presentation.
Thank you, Christy. So Dr. Wiranatne and I worked together in building this presentation, which illustrates how clinical decision solution support software, such as the QIAGEN platform that we will discuss today, allow laboratory to address challenge in genomics. So without further intro, let's get started. So breast cancer is a complex, heterogeneous disease, which subtype characterized by distinct clinical feature and molecular alteration. Advanced NGS have presented unique treatment opportunity beyond stratification of patient by hormone receptor and epidermal growth factor receptor status. So during our presentation today, we will demonstrate how tools like QCI can help laboratory in oncology to select, to look into different cases. So the case that we have selected here, both from a somatic standpoint and a hereditary standpoint, have been selected to allow you to see the utility of QCI Interpret in identifying molecular alteration, seeing how treatment and clinical trial decision support, can support decision functionality, and then demonstrate the richness of the QIAGEN knowledge base. But before diving into actual information about the platform and the clinical case, I have to say that QCI, or QIAGEN Clinical Insight, is an evidence-based decision support software intended as an aid in the interpretation of variant observed in the genomics and NGS data. It is not intended as a primary diagnosis tool by physician. That said, though, it leads me to discuss first about all the challenge that you can have in clinical genomic testing. So if you think about the laboratory performing tests, one of the main challenges that they have is about the scalability in accurate interpretation and reporting. So not only you need to be able to produce a lot of tests, but you also need to be able to do that at a high scale with accurate reporting and accurate interpretation every single time, because the goal of a clinical laboratory is really to be able to provide information to the oncologist and so forth, so better treatment can be done. So you really need a platform that will allow you to balance the risk of the interpretation and the reporting, and as well as expanding essentially the test menu. So you want, as a clinical laboratory, to be able to offer as many tests as possible, so the oncologist and the team of clinicians working with your laboratory can really get to the better outcome for their patient. And that leads to also, from the laboratory perspective, you also want to be able to be operationally efficient. You want to reduce the turnaround time, because sometimes you may be in a really critical situation, or the clinician team may be in critical situation for their patient. So the turnaround time is really important. And while making sure that you're keeping your costs as a laboratory down. So this is three of the challenge. And then the last one, and probably the most important from a human being perspective, is being able to continue learning on each of the cases and provide accurate clinical finding on every single day. So if there is new information, new research, new drug that are approved, or new clinical treatment that are now open, you want to be able to provide that. So you also have to balance your choice of supporting software in a way that you can continue learning about the specific case. So when you think about the workflow of a laboratory, the end goal of many of the laboratory that perform this clinical testing is often the generation of what we call an actionable report, a report that the clinical team, the clinician, and the oncologist can take, and then make the better decision for the patient. So you have to have a report that would allow the lab director, allow the variant scientists in the clinical laboratory to perform information assessment on the variant, add different interpretation about the variant, about the case in itself, when appropriate, when the situation is appropriate, add different treatment and clinical trial. And that today, throughout the use case, the 5 use cases that we will take a look at, that is the end goal, is to put yourself in the place of a clinical laboratory, and how platform like QCI can be used to essentially answer the 3 basic but important questions, such as, is the variant that is in that data set real? Is it meaningful? Is it actionable? So in today's presentation, throughout these 5 case studies, as I mentioned, in breast cancer, we will use that to illustrate to you that breadth of knowledge of QCI and how QCI can be used to help answering these different questions. But before diving into the case study itself, it is important to realize that clinical decision support tools are only as good as the information that is underneath. So you need to be able to have accurate information, and that information needs to be built on a strong foundation. And this is what we are doing at QIAGEN. For more than 2 decades, we have been into the business throughout the different acquisition that we have done, into content acquisition in a lot of different spaces, as well as making sure that all that content, from the primary literature curation, from the integration to different databases, is QC'd and organized in a way that allow you to connect the dots. Because one could say we have 5 million, 10 million, 20, 30, 40 different million findings. If the findings are not organized together in a rich ontology, it is really difficult to be able to take that piece of information and find something meaningful, find an insight for it. And this is what the QIAGEN platform that is underneath QCI Interpret is all about. It's about rich content acquisition, rich and robust QC and enterprise solution, as well as an ontology that allow to connect different pieces of information together, allow to connect patient phenotype with disease mechanism or molecular mechanism, and so on. So all of that, QIAGEN has been doing it with 2 decades. And now, without further delaying it, let's take a look at this platform, but using the different case study that we have. So the case study we will look at today, because it's the Breast Cancer Awareness Month, we decided to focus them on breast cancer. Breast cancer is an important disease. There is, roughly 1 in 9 women in the United States that might be touched, or 12% of the women will get breast cancer at some point in their life. 250,000 new cases in the United States only. Now, the survival rate is great after 5 years, 90% of them we survive, but we need to remember that breast cancer can quickly metastasize in other part of the body. So it's really important to be able to provide the oncologist and all the medical team around the patient a lot of up-to-date information. If we think about two main types of breast cancer, they're based on their location, and typically people will hear about breast cancer that will express different biomarkers. So the biomarkers in the case of breast cancer are HER2 or RBB2, estrogen and progesterone protein. And these biomarker, at least the expression of these biomarker, is currently using as a targeted therapy, right? So you have targeted therapy that will target the RBB2 or the estrogen with tamoxifen-like drug and so forth. However, approximately 20% of the patient are triple negative for these biomarkers. So there's not a lot of things that can be done for this patient. Which, taking all of that into account, we then have decided to select 5 different case studies that can allow you to see the breadth of information and knowledge, and how QCI can really allow you to have a test menu that will focus on all that different etiology of breast cancer. So here we chose from the TCGA databases 2 cases that are somatic, where the driver mutation is in something like BRCA1 or 2. It's actually a more PIK3CA that is the driver mutation in this particular case. We also decided to look at 2 hereditary cases, one with BRCA1 and one with BRCA2, to illustrate also the differences sometimes even in ethnicity or in demographic between the 2 type of gene. And then finally, we couldn't stop ourselves but also include triple negative cases, and in this particular case the driving mutation was MAP2 kinase. So we hope that by showing you these 5 different cases, you can see that a tool like QCI can be used both in the knowledge on the clinical trial and treatment point of view, allow you to have a breadth of information for different cases. Because breast cancer is a disease that the molecular heterogeneity is large, and the QIAGEN solution will essentially allow your lab to be able to address that variety of test menu. So what I will do during the presentation, I will start with the first case, and during the first case we will take a look at the software itself, and then for the remaining of the case we will only take some snapshot, given the fact that we wanted to be able to present to you 5 different cases. So as I mentioned at the beginning, the first 2 cases that we will take a look at are somatic cases. So in this first one, we are talking about a 43 years old Asian female, and at the time she was presented with stage 2A breast invasive ductal carcinoma. She was HER2 negative, PR, and estrogen receptor positive. She had BRCA1 and 2 variant, so she was positive for both BRCA1 and 2. That being said, though, PIK3CA is most likely the driver mutation in this particular case. One other fact that is important to notice for this particular case is that at the time of presentation, this particular patient was premenopausal. She was not in post-menopausal situation. So what I will do, I will go and take a look and dive into the actual molecular information of this particular case. And this is what we see here. So this is QIAGEN Clinical Insight. It's a web application that allows essentially clinical laboratory to submit both the metadata in the variant call file, so that file that contains all the specific variant that I've passed the different quality threshold after sequencing, and essentially wrap information. So within a few minutes, you essentially, after submission, you will get this dashboard view, which will identify for the variant scientist or the lab director in the laboratory, will identify all the variants that are meaningful for that particular cases. So here we can see 5 different variants, and what will strike you is to see the different color here. So you have 2 of the variant, the BRCA1 and the BRCA2 variant, that are color-coded in gray. And gray in this interface mean variant of unknown significance. The other color is red for pathogenic variants. So essentially what QCI is doing is taking all the information that we have in the underneath knowledge base and apply different rule based on different guidelines, such as HCMG guideline, and we classify every single variant based on that computed information. So we can see here the 3 of them that are pathogenic. We will take a look at PIK3CA for interest of time in this particular cases, and 2 of them, the BRCA, are variant of unknown significance. So there's not enough information, or the information is not pathogenic enough to be able to say that these are the driver mutation. In the dashboard here, you also have a lot of few other information. For someone, then Dr. Rathney, my colleague, you may already look at this and know exactly what you want to report out and bring to the attention of the laboratory director. But when you take a look at all the information, sometimes you may have cases that are less familiar to you. Sometimes you may have less knowledge. So the interface allows you to see a lot of different information that you can consider in the decision of whether or not a particular variant should be reported. So you have information such as what is the variant, what is the protein variant, what is the somatic frequency of that gene. So we can see that PIK3CA, this variant has a frequency of 1.24, and this is based on cosmic the bases. So every little I within the interface will essentially bring you to that piece of information more. You can see population frequency at large, impact of the variant, age of onset, and what have you. So you see some glimpse of information of the variant that you have highlighted, which is in this case PIK3CA. And then in gray at the top, then you see information, the metadata or the demographic around the particular case. So it's again, if 43 is all female with ethnicity of Asian and a diagnosis of breast cancer. So if you are someone like me that have to go and dive into the detail, if your goal is also to bring actionability into your report, you want to take a look at the specific variant detail. And that is the second section of the software. So if I click on the variant detail section, that will lead me to essentially a window that will contain the entire aggregation of all the information that we have in the knowledge base. So if you remember one of my first slides where I said, we do curation with QC, we organize. That organization of the knowledge allow us to present the information in such a way that is meaningful for a variant scientist or laboratory director. And if we take a look at the middle of my screen here, we can see, for example, that this variant is a missense variant. It is present in the chromosome 3, and this is the exact location. There is the exon information. There is the translation of that variant in a protein sequence side of the story. And then there is what this variant is about. Is this variant causing a gain of function, loss of function? In this particular case, the variant is causing a gain of function. And again, I can take a look as a variant scientist and laboratory director into why is that there is a gain of function. So in this particular case, the gain of function is based on information coming from the literature, evidences. One important thing that is nice to see with every cases that you may look at is whether or not the variant is in the hotspot region. And that is what we can see here. We can see the variant is in this location here of exon 10. We can see the little red information here, which represent piece of information in the QIAGEN knowledge base indicating that these variants are pathogenic. So we can see that indeed this variant is within the hotspot region, and the yellow domain indicate to us that it is in a critical domain of the protein. So this variant is indeed in a hotspot region, accumulating a lot of variant, pathogenic variant, and is in a critical domain and causing a gain of function of the protein. So From there, depending of the workflow of the laboratory, depending of what is the goal of the report, whether you report clinical treatment and trial and so forth, there's a lot of other information that you can take of. So if, for example, the goal is to provide clinical trial and treatment information, then you have this information in the next section of the interface. And the treatment and trial information is divided to you from a really, really narrow view to a really, really large view, where you can see essentially treatment and trial that are related to the exact variant, to the entire gene, by simply clicking like I'm doing right now and looking into expanding from exact variant to the gene positive. Likely, you can narrow to look into treatment and trial that are approved for a specific indication, such as breast cancer in this use case, or any type of cancer. So sometimes you may have no other choice but to look at whether or not the treatment in the trial is available in all cancers. So this is also something that you can see what you can do with this interface. And if we look at the information here, what my colleague has done is to take a look at what is the information that we have in terms of clinical trial. So the first one that I want to bring to your attention is the first one. It's the first trial that we have here. So if I look at a treatment, it's a combo treatment where you have two different treatments. The evidence in the literature is coming from PIK3CA mutation. Yes, we do have one. ERBB2 expression negative. Yes, we do know that it's negative. Estrogen receptor positive. Yes, we do know that. So it's looking good. In this particular case, though, the treatment, the clinical trial, is open for postmenopausal women. So for this particular case, unfortunately, it will not be recommended to put on a report, for example, to put this clinical trial on the report. This is something that the lab director would most likely not recommend. And because of that, you can see here in the icon here that there is a big red line striking through, meaning that this clinical trial is not reported into the report. Even though it sounds promising, it will not be appropriate for this particular case. But when you take a look at all the other clinical trials that there is a lot of them. I'm not going to take a look at all of them, obviously, today. That some of those clinical trials are in dark. They are, again, against the PIK3CA inhibitor, targeted. Some of them are relevant. For example, this one here, where you have HER2 negative again. There's different state or worldwide location when they are outside of the United States. You always have the phase of the trial. So one thing to take into account is all of these trials are currently enrolling. So they will accept new patients. And then you have the clinical trial record that you can always link out and see. And my colleague has done all the exercise of looking into that and identify potential clinical trials that are meaningful for this particular case. So once, when that is done, depending on what is needed, you may, the variant scientist or the clinical laboratory may stop here, or you may also be interested in looking into the literature evidence. And what we can see here in my screen is all the piece of information that the QIAGEN knowledge base underneath this tool is having. So this is all curation that we have done that the QIAGEN curator have read and then put in the knowledge base. And some of them are about somatics. So you can see a vast majority of the information in the knowledge base is around PIK3CA as this particular variant, as a somatic cases. And then some of them, though, are coming from inherited. So they are individual related and most likely from inherited example. And the fact that they have a darker color and they're not red means that they are concordant with this particular case that we are looking at. So you can feel confident that this variant is indeed, of other cases, somatic, is a somatic driver variant in a lot of other cancer. So if you look at the bibliography, if the goal is also to dive into specific article, you can then open this view and essentially see all the publication. And as you can see, this was actually, we have worked on that case study a few weeks ago, so this was not there at the time, but you can see that QIAGEN every single week will bring you the most up-to-date information. So we can see here a publication that was published in mid-September, EPUB in mid-September, where this variant has been shown in cases with metastatic breast cancer or primary breast cancer. There was also a lot of other publication that we can dive into and look into the more specific information as well. And some of these publications, when it's known the ethnicity, will also be shown. And this is important because clinical lab, in some cases, depending, will also want to report out main key publication about the variant. And this is what this view allow the variant scientists and the laboratory director to do. And then finally, once all the information has been reviewed, and it's the time for the variant scientists and the laboratory to produce the report, they can simply go into the review and report section and essentially annotate and produce on the spot their report. So they can add overall interpretation on the case, discuss about the clinical trial that were identified, discuss about the consequences of the different variant. For example, here note that the BRCA variant were not, their clinical significance is not established, and talk about the different targeted therapy or clinical trial. And then for each of these particular variant, the variant scientists can also type in or use in the interpretive comment and discuss about the impact of these variant within the particular case. And when appropriate, clinical trial and treatment can be added to the report as well as the references. And if you are a lab director, you can sign out the case right there and then produce the report, or you can simply preview the report and generate a branded PDF that would allow you to communicate either with the tumor board or communicate with the oncologist team and so forth. So you have that report that will contain demographic information about the specific case, the overall interpretation, summary table, as well as the information for all the variants and treatment that are meaningful to report in the given case. So what I will do now, I will go back to my Excel, and in a nutshell, if we think about the clinical case itself for this particular case, what we have seen by using clinical QCI Interpret, we've seen that the BRCA1 and 2 mutation are not pathogenic. They have a unsignificant, so they're variant of unknown significance. We have seen that PIK3CA is an activating mutation in the hotspot region. The particular cases is eligible for different PIK3CA inhibitor. There is also TP53 variant that are important in this particular case, and we have not dived into P53, but if we were to look at the information, you will find that some of the variant that we have is eligible for different clinical trial, including the WEE1 inhibitor trial. Now, when we were working at that case, I asked my colleague, I said, but what about male breast cancer? Is there anything in the etiology of the disease that is similar, and so forth? So we chose the next case study as actually a male cases. So it's a 68-year-old Hispanic gentleman that was presented with stage 2 B liminal invasive ductal carcinoma. He was HER2 negative, ER and PR positive. In this particular case, BRCA1 and 2 was negative. So as some of you may know, male breast cancer cases are very rare. So if we look at the United States, for example, there's about only 2,000 patients per year that are newly diagnosed. But we can see based on this, and you will see when I will look at the molecular etiology of breast cancer transcend gender. So some of the variants are the same, and this is the case here. We see a different variant. You will see different variant than in the previous case, but it's still targeting the same genes, still targeting PIK3CA. So if we take a look at the case itself, when you look into the interface, you will see that the PIK3CA is indeed a pathogenic variant. So this is a different variant in this case. Its somatic frequency, though, is higher than the previous case. So we are looking at the 3% somatic frequency. So it's a more well-established, I would say, variant in cancer at large. Population frequency is really low as well, based on exact. And the differences between the PIK3CA here and the other one is really about treatment. Seems like there is potential treatment option for this particular case in PIK3CA. And if you dive into the knowledge, you see something a little bit different. You see also rich information. It's a well-established, well-published, documented variant in the literature. But you can see, I did not count them, but you can see that it's not half and half, but almost half and half of the information is either from an inherited situation or from a somatic situation in this case. And if you look at the clinical trial, we're looking at something pretty similar to the previous case that we have seen. But in this case, we could almost say that the clinical trial that was the first one that was targeted for post-menopausal women could be something that the lab director and the clinician may want to talk about with the clinical trial leader and see whether they could enroll this particular patient in the clinical trial. Even though it's for women, there is, this particular case is also ER positive, HER2 negative, so it's possible that they will accept this particular case within that clinical trial. So the interface allows you to see that clinical trial, and it is a meaningful trial in this particular case. Male breast cancer are very rare. We can notice the same disease etiology in the case of breast cancer. We have a different PIK3CA variant, but it is still an activating mutation in a hotspot region. And it is also a variant that is eligible for clinical trial. Now, we have seen two different cases of somatic cases where PIK3CA, one woman, one male. And what we thought we would do for the next two is to look into more classical, or what people will think is the more classic cases of breast cancer, where in this case we're going to look at two different inherited breast cancer cases, one in BRCA1 and one in BRCA2. So the first one is a case in BRCA1. So BRCA1 variant, typically they are more present, more seen in women that are not Caucasian. So here, if we take a look at this particular case, we have a 33 years old African-American woman that was presented with a stage 2B infiltrating ductal carcinoma in the left upper quadrant. She was ER2 negative as well, PR-ER positive. She was positive for BRCA1, and in this, based on the variant profile, the BRCA1 is most likely the driver mutation in this particular case. She was premenopausal. So if we take a look at the actual information in QCI, we can see that the computed classification is coming in with indeed a pathogenic classification for this particular case, where you can see the BRCA variant, you can see other different variants in this case that are variant of unknown significance, and that is why they are in gray here. And if we dive into the information, we can access now the computed classification detail. And in case of inherited cases, what you see, what is recommended in the guideline is using the ACMG guideline. And this is what QIAGEN Clinical Insight does. So QIAGEN Clinical Insight look at the information in the knowledge base, and we have computed all the different guideline in the ACMG guide, and that's what you can see here. You can see at your fingertip the different rules that are turned on, the different criteria, the strength of the different rule, why. And then all those rules are contributing to this classification that is pathogenic. So here we're talking about the null variants of frame shift information, which is a very strong, absent in different controls, such as exactas and genome prop and so forth, and containing a lot of computational evidences that support deleterious effect. Now, one of the questions we had is we started with the assumption, okay, so BRCA1 is more present in cases that are not Caucasian, and here we are. We have a case that is with a demographic of African Americans. So, what about the literature? And when we took a look at the literature, we had one publication. So really, all the curation and systematic curation, the way QIAGEN does, allow you to identify even those rare publications. And you have one in there. And when we took a look at it, we actually saw that it was a publication in 2015 where BRCA1 and BRCA2 was studied in the Lebanese Arabic woman that had high hereditary risk of developing breast cancer. And we can see that this particular variant, this particular BRCA1 germline variant, heterozygous variant, was observed in these cases, in these Lebanese cases, with familial breast cancer. So it was comforting to see that not only this particular case had it, but there is other, women with different ethnic background that were also having this inherited potential variant that developed, with familial breast cancer. If you think about BRCA1, BRCA2, sometimes there is potential of going into different treatment and different trials. So what we can see here, take a look at the treatment and trial, you see that there's not really approved just yet based on NCCN, ESMO, FDA, EMEA guideline. There's not a lot of different drugs that are approved. You have a drug for the platinum chemotherapy standard for triple negative, but there's nothing really for BRCA1 and BRCA2. Now, there is something in ovarian cancer that is approved for olaparib, and that's what we can see here, where we can see that these second-generation PARP inhibitor could be approved as off-label. So if the laboratory director and the clinical team decide to go that route, they could go and ask for a particular permission to be able to put this patient on the olaparib. But if we take a look at the clinical trial, actually, we can see that there's currently in the United States in phase 3 a large olaparib clinical trial for breast cancer in this case, where you have the case where the woman is R2 negative, BRCA1 and 2. So it's really a clinical trial that this patient is with olaparib, and it's a clinical trial that this patient could be enrolled today. So this is a nice example of a clinical trial that could be added to the report as well if the clinical team decide that it's not appropriate to go in an off-label situation for this case. But you still have hope because there is clinical trials that are open. So in summary, we can see that you can see and access different FDA-approved information such as olaparib in an off-label setting for this particular case. But you can also look into second-generation PARP inhibitor as a clinical trial, and I mentioned olaparib, but there's a few other clinical trials in this particular case. So if we now we took a look at the hereditary case, we took one example of inherited BRCA1. So what about BRCA2? So if we go and we take a look at the BRCA2 cases, here we have a 51-year-old Caucasian female was presented with a stage 3C breast cancer. She was HER2 negative again, ER and PR positive, with germline BRCA2 variant, sorry, and she was premenopausal at the time of the biopsy. So if we dive into this specific information and look at it, we can see that in the opposite, all the other cases that we've seen so far were pathogenic. So in this particular case, the BRCA2 variant, the information that, based on the ACMG guideline, the information that is available in the knowledge base based on the different ACMG rule, don't call this variant as a pathogenic for breast cancer, but call it as likely path. So it's still something that is a variant that is still of interest to report out. But you can see how the rule can really differentiate subtle change of information, where you see here, variant of unknown significance, pathogenic, and likely pathogenic variant. And when we dive into the assessment, we can see that the rule are a tiny bit different based on, compared to the one before. We're still having an effect of a frame shift, a null variant in this case, absence from low frequency, absence from different control, and there's multiple lane of computational evidence as well, but overall, all of that combined together for this particular variant makes it as a likely pathogenic cases and not a pathogenic case. If we look into treatment and trial, well, we're going to get into a similar situation in the BRCA one, where we could always, the laboratory director and the practitioner can always go and look into olaparib situation off-label, or look into the different clinical trial that may be available, such as the olaparib, as I mentioned earlier, but also that telasuparib, that is another clinical trial in phase 3 related to the second generation PARP inhibitor. So similar to the BRCA1 cases, this case is illustrating that you can use the ACMG classification in an automated way to differentiate the pathogenicity of a variant, but also still access a breadth of information with regards to treatment and trial when the need is. The conclusion is pretty similar in this case, but it's just to illustrate how ACMG classification can really differ from case to cases. And finally, as I mentioned at the early beginning, we couldn't stop ourselves but ask the question about what about triple negative? So what QCI can provide to you when you look into a triple negative breast cancer cases. So the last cases of today's presentation is a triple negative breast cancer. It's really challenging to treat because the tumor do not have HER2, ER, or PR. So they cannot, these particular patients cannot go to a standard treatment, HER2 inhibitor, or tamoxifen, and so forth. They have to have something else. And since we mentioned at the beginning that there's about 50 to 20% of breast cancer that are triple negative, we thought it would be interesting to take a look at this. So the case is the case of a 50-year-old woman, Caucasian, presenting at the time with breast invasive ductal carcinoma of the left lower outer quadrant in this particular case. There was a lot of different variation. BRCA1 and 2 was negative. MAP2 kinase, there was a MAP2 kinase variant in an outspot situation that was also present there. And then there was a lot loss of function in the P53. So if we take a look at QCI, again, in these cases, what we will see is two pathogenic variants, so the P53 and the MAP2 kinase variant. So we can see that this MAP2 kinase is pathogenic cases. It has a really low somatic frequency in this particular case, but if we start and dive into the detail of this, we can see a lot of useful information. First of all, the MAP2 kinase is a case where there is different treatment, not for breast cancer, for melanoma, but there's different treatment, different variants, such as this exact variant actually can confer resistance for different drugs. So it might be interesting for the laboratory to know if the lab, if the oncologist team were thinking about going in the off-label situation, knowing that this particular variant is causing resistance in some cases, might be of interest for them. But there's also two other for which the treatment might be or approved for melanoma, and so by the FDA and EMEA, and could potentially be of interest. And if we go into the clinical trial, one of those drugs, or actually both of those drugs that are for melanoma are now in clinical trial phase 2 this time in a few different states in the United States for breast cancer. So we can see here MAP2 kinase. It's a combination where this drug will be in combination with this standard triple negative case as a first-line treatment. So it is interesting for the laboratory to know and to provide the information to the clinical team that there is a new clinical trial, correctly, really new. It's still in phase 2, but in some stage for triple negative, that a combination of an established treatment for melanoma is currently being tested in triple negative breast cancer in the hope that it will help. So this is again information because you have access to richness of the clinical trial treatment information and up-to-date information the lab director can provide. So without, in the interest of time, without diving into the detail, we can see that the MAP2 kinase activating mutation has different FDA-approved treatment, but there would be off-label, so it would mean that the clinical team will have to go and ask different permission to the different agency. But there's also clinical trial, and there's clinical trial as well into the P53 situation. So in summary, our goal was to use these 5 different cases to show you that clinical decision support platform can be used to really classify, annotate, and provide rich information and report out for the lab director and the variant scientist. It provides color-coded information in the form of a dashboard that allow the team to quickly see what is pathogenic or not pathogenic. It provides access to up-to-date libraries. We've seen publication, for example, that were published in September of this year. You have also access treatment trial, depending of the case, that might be meaningful for the patient. And obviously for the laboratory director, they also have an interface that they can use to enter their comment, both on the variant level, treatment and trial, as well as the case level, so they can provide and generate a report to send out to the clinical team. So in conclusion, clinical decision support is key in precision medicine and in oncology. And by staying current with the QIAGEN knowledge base, which is the most trusted source of clinical and biological finding, clinical reporting, it will help the team, obviously. It also helped the laboratory to increase the throughput of your clinical genetics team and the variant analysis capability of interpretation by having access to breadth of information. You can stay current with the industry-leading knowledge that are accurate, up-to-date, based on the content is coming from a lot of different sources based on scientific, clinical, therapeutic, and prognostic evidences. It improves also your decision-making by automatically applying relevant professional guidelines and variant classification rules to deliver actionable evidence-based clinical insight for the different patients. Get the maximum flexibility of integration and customization, so you can customize this to enter in your workflow and your specific reporting rule. And finally, develop a private lab-specific knowledge base. So what I would like is to thank you all, and now you can stay here, and we will give you the opportunity to discuss with me or with Dr. Veratnatni that have worked into that case for today's presentation. Thank you, and have a great rest of the day.
Thank you so much, Dr. Duchenne. And now we will open up questions to all attendees for both Dr. Duchenne and Dr. Veratnatni. So please feel free to enter your questions in the questions box of the control panel. Just to get us started, thank you both again for the interesting case studies, and I wanted to just mention the triple negative breast cancer case studies really seems like a great example of how precision medicine will help in the clinic. Dr. Veratnatni, could you comment on that?
Yes. So so this is Dr. Dilhan Veratnam. Yes, that triple negative breast cancer case was a classic example of targeted therapy and precision oncology, which could help out in the clinic. as you know, triple negative, as Dr. Deschamps mentioned, triple negative breast cancer is particularly hard, challenging to treat. So typically in the clinic, chemotherapy regimens like anthracyclines, like doxorubicin or epirubicin, or taxanes like paclitaxel and docetaxel is used to treat TNBC cases. However, the challenge with this is that oftentimes there are a lot of grade 3 and grade 4 toxicities with some of these chemotherapy agents. A case in point is like doxorubicin, a chemotherapy that is used to treat these cases. Many patients have cardiac complications. So, and oftentimes, as is in the case with this patient, there will be recurrence of the cancer. So if it's a triple negative breast cancer case and the oncologist decide to do NGS sequencing, most likely that the patient have exhausted the first, second, or even third line of chemotherapy options. So at this point in time, the oncologist is left without any recourse. Now, in this case, I'm pretty sure the oncologist would have been extremely excited to see the sequencing results of this patient, because this is a clear-cut example that the patient has a MAP2K1 mutation, and there are targeted therapy approved for melanoma. As Dr. Deschamps mentioned, cobimetinib and trametinib is approved for malignant melanoma. So what the option the physician would have here would be to write a letter of medical necessity to the insurance provider, get prior authorization from the insurance provider, and the content in QCI Interpret could be used to substantiate the argument. So this so as you can imagine, trametinib or cobimetinib is very expensive if you are paying out of pocket. It's about $14,000 per month. So so you could prescribe it off label if you can get a prior authorization from the insurance company. Alternately, the patient could be enrolled in a trial. And as you saw, there are plenty of promising clinical trials, including cobimetinib, trametinib, and there were a couple of trials with third generation MEK inhibitors like selumetinib. So this was a poster child, basically, of how targeted therapy will help a patient who otherwise would have had no options in the clinic.
Thank you very much. From a variant scientist or clinical geneticist point of view, how would QCI Interpret help a clinical lab?
Oh, that's a great question. So predominant majority of the time spent by variant scientists is on the literature curation and variant interpretation. An internal study we did at QIAGEN, we have shown that QCI Interpret reduces the test interpretation time by almost 75%. So that time that is saved can be used for secondary review, QA and compliance, and also scalability. So other clinical decision support products that are out there, like you annotate variants using sources like HGMD and ClinVar. But the problem with that is they lack curated content for primary literature. So this is especially problematic if the variant interpretation is tied with the ACMG classification. Now, QCI, as Dr. Deschamps mentioned a couple of times in her talk, we have a very robust knowledge base curated by our expert PhD level scientists. So this includes findings from primary literature, clinical guidelines, FDA drug labels, multiple trial registries, not only the clinicaltrials.gov, multiple trial registries around the world. And I want to point out the other definitive advantage of using QIAGEN Clinical Insight as a clinical decision support system is that we assess the entire molecular profile of the patient in its entirety instead of the alternate approach of variant by variant analysis. So when we do recommendations for treatment and trials, we look at the combination alterations, so multivariants, as you would say. So the final recommendations we give would be based on the entire profile of the patient.
Excellent. Thank you very much for that. And I think we'll have time for just one more question. So just one more question here. For genes that have multiple transcripts, which transcript do you represent in QCI Interpret? And do you have information on the lesser known transcripts?
Oh, that's a great question. For genes that have so for the genes that have multiple transcripts like FGFR1 or GNAS or many other genes for that matter, what QCI does is they use the transcript with the largest number of findings in the knowledge base as the default transcript. So if there are two transcripts with the same amount of information in the knowledge base, then the QCI assigns the longer transcripts as the default transcript. However, all that being said, all the rest of the transcript information is available in the application. FGFR1, for example, has nine different transcripts, and you can see all these 9 different transcripts in the variant detail page. And if you are interested in some other transcript other than the default transcript, you can toggle between the 9 transcripts and select the one you would wish to analyze. So all the information is pertinent on the transcript you have selected.
Wonderful. Thank you very much for that. I think that was the last question that we have time to address for during today's webinar. Thank you again, Dr. Dushen and Dr. Wiratne, for your exciting presentation. And thank you to all the participants for joining our webinar and submitting great questions. To find out more about other QIAGEN bioinformatics products, please feel free to visit us at QIAGENbioinformatics.com. And this will conclude our webinar. Thank you very much, everyone, again for attending, and have a wonderful day.